So this is our third neuroscience lecture. Obviously, the first two were connected by our other tutors. This one is my lecture. Briefly, from what I understand, your previous lectures were about what brain regions exist and roughly what a neuron is made of, right? Like, what did you discuss in previous lectures? Lobes, okay. Yeah, Etna, you can speak without raising your arm. Yeah, you can just speak. Okay, yeah, lobes. What else did you discuss?
I wanted to say the same, lobes.
All right. I believe that was the first lecture, right? And what was in the second lecture? Maybe some neuron.
It was the history of neuroscience.
Hmm. Okay. All right. In that case, I'll start today's lecture with a bit about basics of what a neuron looks like and how it's made and so on. And then I'll go into today's theme a bit more. Okay. So you already know that our brain is made of different lobes. So there's frontal lobe here, which is mostly for consciousness, language, and intentional movements. There's parietal lobe here, which is mostly for senses and spatial awareness. There's temporal lobe to the sides here, which is mostly just hearing and understanding language. And at the back, we have the occipital lobe, which is our vision center. So it's where we understand what we're looking at, and also the part that is stimulated so that we can see things in dreams and hallucinations as well. That's the basic idea. Now, to be clear, more accurately, we could say that our nervous system, which is the entirety of, let's say, our thinking organ, is made of central nervous system and peripheral nervous system. So peripheral is just nerves that go into your arms, legs, and just outside, so that they are basically sensory neurons that let you feel touch, temperature, pain, and so on, and also motor neurons that let you control your muscles to move your hands or to speak and such. And there's the central nervous system, which is our brain and our spinal cord. So spinal cord is just basically part of nervous system that goes through our spine, and it connects to all our muscles and just all of our body that we control or interact with. But mostly neuroscience focuses on the brain because that's where you could say more complicated things happen. So in our brain, we have, roughly speaking, two main sections, the cerebrum and the cerebellum. The cerebrum is typically what we call the brain, which is what's made of these lobes, so frontal lobe, parietal, temporal, and occipital. And the cerebellum is something we have at the back of our heads, which is like our navigation center. That's what gets messed up if you drink too much, which is why you can't walk straight. Um, so obviously, for the most part, we focus on cerebrum, which is where the lobes are. But everything that we say about neurons generally applies to all of our nervous system. So it applies to the nerves we have in our hands, in our legs, just everywhere. So today, our discussion will be more broadly about how a neuron works and how neurons communicate with each other. So at first, what we need to understand is that our entire body is basically made of three kinds of neurons. There's sensory neurons, which take information from the outside world, like we have neurons in our eyes that let us, well, see color and understand that I'm looking at this person or that I'm looking at this object. We have neurons in our nose that let us smell. We have neurons in our tongue that let us feel taste, for instance. And all over our skin, we have sense of touch, pain, and temperature. We also have some sense of hearing, balance, and a lot of other things. But basically, these are all sensory neurons, which means that they get information from the outside world and then they give this information to other neurons. We also have motor neurons, which is neurons that control our muscles. So for instance, when I am moving my hand like this is controlled by a lot of neurons because each kind of different rotation that I perform here has to be controlled by a group of neurons because it's a group of muscle fibers being moved in this way. So everything that we actually do in the world, we do by motor neurons. So walking, moving our hands, or even speaking are all done by our motor neurons. So we sense the world with our sensory neurons, and we act, move, use our hands, speak, or whatever with our motor neurons. Even like our blinking and swallowing and just everything that we do is with motor neurons. And until our discovery mm, of the ability to use brain-computer interfaces, pretty much you had to use your muscles to communicate. These days, that's not the case anymore because we can directly read our brain signals to maybe send a message, which is, for instance, what Neuralink patients use to telepathically send texts and play video games. We'll cover how that works in our week 6, the last week. But for now, let's just stick with the basics, which is that we have sensory neurons that take information from the outside world and give this information to other neurons. We have motor neurons that receive an order from other neurons and then they make our muscles move in a certain way. And in between, we have interneurons. So interneurons are just neurons that receive information from some neurons and give information to other neurons. So for instance, our brain, as in our cerebrum, is mostly interneurons. That means that these are neurons that process some kind of information, they do some kind of thinking, reasoning, feeling, whatever, so that in the end they will decide on what to move, what to say, and just what to do with motor neurons, basically. So sensory neurons are input, motor neurons are output, and interneurons are everything else. Now, today we will discuss how these neurons actually communicate with each other. And yeah, basically that's where spikes and synapses and neurotransmitters come in, which is going to be the main theme of today. So let me share my screen and we'll begin. And if you get any question throughout the lectures, or if you prefer a different analogy, different explanation, or more visuals somewhere, just stop me and we'll do that. Okay. So mostly we'll be talking about signal transmission and synaptic logic today. And basically signal transmission just means how neurons talk to each other, like how they decide that if I see a car coming at me, I should move away. And synaptic logic is just how neurons connect with each other. So let's just refresh on the main ideas. So suppose that this, what you see on the screen, by the way, do you see the screen?
Yes.
Okay. So this that you see on the screen is a typical shape of one neuron. So a neuron is made of a cell body right here, which is called the soma, which is where regular cell stuff is. Like all cells in humans have DNA, they have ribosomes, they have cell membrane, which is like border that protects them from the outside world. And they have all sorts of processes going on inside them. They're made of water, they have all the proteins that do different things, just everything. And neurons are also cells, so they also have a cell body. But what makes neurons different from other cells is these two structures. So this one is called axon, and these are called dendrites. So axon is what a neuron uses to send out a signal to either another neuron or to a muscle. So for instance, if my eyes just received light, like because there's light in my room, my eyes will activate and the neurons that I have in my eyes will send a signal through their axon, which will be connected to another neuron, and that other neuron will maybe say, oh, my eyes hurt, I should look away, or something like that. So the basic idea is we have many neurons in our body, and they send signals through each other, and the way they do it is the neuron always sends out a signal from the axon and receives signals from the dendrites. So any neuron can have one axon that branches out, but it's one axon, meaning that it sends everywhere at the same time. And it can have many dendrites, which receive from all the other different places, like different neurons. So for instance, this one neuron might be receiving signals from, let's say, 5 different neurons, one here, one here, one here, and so on. And for each signal, basically what that means is that there will be another neuron here whose axon will be connected to this neuron's dendrite. And that connection is called the synapse, and that's what we'll be discussing mostly today. Now, before we go into how neurons connect to each other, let's briefly discuss what we mean when we say that a neuron sends out a signal. So let's get the intuition for it. First, this is neuron's axon, which it uses to send a signal, and it has a start point and an end point. The start point is right here. It's called an axon hillock. It just means axon's beginning. And endpoint is right here. It's called axon terminal, so where it ends. The idea is that at the axon terminal, we'll have dendrites of other neurons which receive the signal that we sent. Now, first of all, some main concepts to just throw out there. Our brain is mostly made of neurons and glia. So neuron is just what we've been discussing all this time, and glia is all the support structure cells that protect neurons. They clean up the waste chemicals around them, and they even speed up the signal transmission in neurons. So for instance, right here on the image, you can see that there's these like red cylinders wrapped around the axon. These are a specific type of glia that create what's called a myelin sheath. Don't worry too much about memorizing this right now, but basically myelin sheath uh, insulates this axon. So the axon has to send an electrical activity. And in order for this electrical activity to not get spread out and lost throughout the body, it's protected by this glia so that it arrives at the destination properly. So just like the cables that we have at home, like even this mouse cable right here, the actual wires that carry the information are inside this, but on the outside we have this support structure. Like if I cut this open, then we will see the wires, but then it won't be safe or it won't transmit messages as well because some of the electricity will leak out. And to make sure that the electricity doesn't leak out, our brain's axons have this myelin sheath around them. Now let's discuss some details of how a neuron sends out the signals. And by the way, if you're texting me something in the chat, let me make it so I can see that. Okay, if anything, I'll check the chat. Oh, yeah. Let's see. Can I turn this here? All right, let's just continue. So, if you get any questions, ask me at any moment. And now I'll briefly discuss how actually neurons decide that it's time to send a signal. And the basic idea is that a neuron has a lot of ion channels. And we'll see what they are in just a second. But first, get the intuition that a neuron has this cell body, and it has to accumulate enough voltage inside by getting enough charged particles. Yes, Atuna?
I have a question. How many neurons does we have? And if one neuron got damaged, does it affect the whole structure?
Okay, good question. Thanks for asking. So it's estimated that an average adult human has about 85 billion neurons in total. In total means in all of your nervous system. But specifically the part of the brain that we're interested in, the lobes, that's like 15 billion. So about 15 billion that goes into our thinking and feeling and that kind of, let's say, high level things. Sorry. But 85 billion in total if you count all the sensory neurons in our skin and all the motor neurons and everything. And it's estimated that each neuron connects to about 1,000 other neurons. So there's about 85 billion neurons and about 100 trillion synapses in our body, in our brain and the spinal cord and everywhere. And it's usually not a big deal if only one neuron is damaged because there's billions of them. But some neurons are very important, so it depends. Like mostly it's fine if one neuron gets damaged because there's usually a group of neurons doing the same job. But if too many in a group get damaged, that's when you might lose hearing or you might lose vision. Or sometimes, for instance, we have a specific brain area here that is only, its only job is to recognize faces. It's called fusiform face area. We'll discuss it. And basically, if enough of your neurons get damaged in that area, you can still recognize objects. You can still recognize color. You can still recognize motion. You can still even read letters, but you can't recognize faces. You can't tell who you're talking to, even though you're capable of, for example, reading a text or other things. So yeah, you could lose some let's say, abilities or features. But generally, if only one or two neurons die, it's fine. Although one thing to consider is that most of our neurons in our brain don't regenerate. So some neurons can regenerate. So if they die, it's fine. Like, for example, our sense of smell can regenerate even if we lose it. But other neurons, like vision and hearing and such, if you lose them, they stay lost. Now, you don't necessarily lose them in the sense of dying. They might just get less powerful, like less sensitive or whatever. And in that case, you could train to get them back. But yeah, if the neurons die, usually we can't recover them. There's some new experimental medicines to fix that. But for now, yeah, if you lose it forever. Which is why it's important for people, even if they retire after their work, to still stay intellectually engaged so that their neurons get enough stimulation to not die out. Which is why actually people who don't retire much, or at least keep an active life after retirement, have less chance of getting Alzheimer, dementia, and that kind of stuff. So basically, yeah, we have 85 billion neurons in total, but 15 billion of these are for our, let's say, thinking side. And you could lose a couple and it won't really damage you significantly. But if you lose too many in one specific area, because our brain is very specialized, like we have one area for reading, one for writing, one for seeing objects, one for this, one for that. And so if too many neurons in one area get damaged, you might lose that ability. Did that answer your question?
Yes, it was clear. Thank you.
Okay. Yeah. If anyone gets any question, feel free to interrupt me similarly. And yeah, for now, let's continue. So the way that a neuron is capable of sending out signals is that it has to build up charge. And in order to build up charge, basically in our body, there's always a flow of ions. Ions just means charged particles. So for example, there's hydrogen, calcium, potassium, whatever, all these different particles that flow in our body. And some of them are positively charged, some of them are negatively charged. And each neuron actually manages how many of each to keep using the ion channels. And basically, in total, this leads to either accumulating a positive charge or accumulating negative charge inside a neuron. And when the charge is positive enough, it will fire a signal. We'll get to the signal in a moment, but let's take a second to understand ion channels. So here's what ion channels mean. This thing right here, you see this orange balls separated by threads. This is called a phospholipid bilayer. This is a wall that every cell has in any body, human body, animal body, plant body, everywhere. There is a phospholipid bilayer, which just means that this is a structure that keeps the outside and inside separate. Because, for example, maybe outside there's water, there's all the chemicals we don't want, but inside the cell, there needs to be an exact concentration of the right chemicals so that correct things can happen. Now, this bilayer keeps almost everything away, but sometimes we need to take up things. For example, every cell needs energy, which is why they need to take up some sugar so that they can break it down and get ATP for their cells in mitochondria. Or every cell needs to get rid of its waste. And for all the different kinds of, let's say, mixing that we need to do between inside the cell and outside the cell, we have these membrane channels. And specifically for neurons, we have channels that are for carrying inside or outside just charged particles, which are known as ions. So these are the ion channels. They can work by different principles, but the idea is always that some charged particles will be allowed to enter a neuron and some charged particles will be allowed to leave the neuron. So that in total, a charge will build up, and when enough charge builds up, the neuron sends out a signal. And eventually, the neuron that receives this signal also sends a signal, then that one also sends a signal, and in the end, we move our muscles and our tongue and our throat and our eyes with them, and basically we interact with the world that way. Now, there are different kinds of ion channels. Let's start with the simplest one, which is ion channels that are always open. So it's just simple ion channels like this, which a specific kind of chemical, like let's say in this one potassium, in this one sodium, there's also ones for hydrogen and ones for chloride and so on. And basically each one can only carry this one kind of chemical either inside or outside. So basically a gradient forms, like if the inside is too much negatively charged, then some of them will flow outside or the other way around. And yeah, basically, so the always open channels allow a specific kind of charged particle to always flow in or out, depending on which side is less concentrated. So basically, it goes from more concentrated side to less concentrated side. There are also other kinds of ion channels which open under specific conditions. For instance, one that is most important is voltage gated. Voltage gated means that when enough charge builds up on the inside of a neuron, some ion channels will open or maybe some will close. So this means that maybe this door is usually closed. So natrium ion, which is just sodium, by default won't be able to, let's say, leave the cell. But if we get to the certain voltage, then it will open and then it will be able, like let's say this is the default case where the channel is blocked, but after reaching a certain amount of charge inside, there will be enough voltage and this will cause it to open up. Similarly, we have mechanically gated ones, which means that, for example, our skin works this way, that if there's enough touch on the skin, whatever was blocking the channel will be moved away, and then ions will be able to flow in. Like in this case, this is about blocking. Let's see. For instance, by default, our skin has some ion channels which are open, and so we can sense things because they keep moving in. But if there's enough touch on some certain direction, they might close, and then we won't be able to receive these chemicals anymore. And there's also ligand gated, which basically just means that a certain kind of chemical closes or opens an ion channel, and this chemical might be sent out by another neuron. So these are just complicated schemes that build up in our body, which together control when our neurons send signals to each other. We'll have some more examples of this in a moment, but this is just a main overview of it. Now let's briefly discuss the spikes and synapses. Yeah, let's just do it. But right now, about ion channels, do you have any specific questions?
No.
Okay. If you get any questions, interrupt me at any moment. Right, so ion channels are basically what allows neuron to build up charge because they allow flow of charged particles. Now, why do we care about charge? We care about charge because there's this thing called spiking. This is a little more dense image, but let's go. Okay, let's start with this actually. So basically, imagine this. This line, so this horizontal line is time, and this vertical line is just neurons' action potential. So the voltage buildup inside the neuron. So let's say that at rest, our voltage is right here. It's about minus 70 millivolts charge inside the neuron. And when we receive certain positively charged ions, our charge starts to build up and go up. Now, eventually, at some point, because our ion channels also make some charged particles leave, the charge starts to come back down. But then we receive some more signal that goes up, but comes back down, and so on. And if it happens that we reach this threshold, the threshold is at minus 55 millivolts. So if our neuron happens to build up enough charge to reach minus 55 millivolts, a spike happens. A spike means that a lot of the ion channels open up at the same time. Because remember, some of them are voltage gated. So if we reach minus 55 millivolts, a lot of them open up at the same time. So we get a lot of a positive charge building up inside. So our potential inside the cell builds up a lot. It happens very quickly. So on the graph, it will look like it's spiked up, which is why it's called spike. It's also called action potential. And when it reaches this high point, this is when a neuron will send out a signal through its axon. And this signal will be received by other neurons. And eventually, the signal will go all the way to the kind of neurons that control muscles. And that's how we move. That's how I speak right now and how we do everything, basically. Now, after we reach this point, the ion channels will begin to slowly close. And as the ion channels close, the neuron will start to lose charge again and come all the way back down. It will undershoot, come too much back down, but then over time it will stabilize and go back to the same voltage that it had before. And after it has returned to the same voltage, then it can be stimulated again and can shoot up again, come back down, and so on. This single process is called a spike, and a spike is like a unit of communication between neurons. This is how they tell each other, I'm hungry, I need to walk, I need to move my hand, I'm thinking about some geometry problem, or I'm feeling sad, or basically everything is encoded by how often and which neurons send out spikes. And to be clear, we don't really understand how come this makes us feel anything as opposed to just simply responding without any feelings. We have no idea how come we feel things, but we know that this is the mechanism for how we communicate inside our brain. So to be more clear, let's bring in some technical terms. So this is called the resting potential. It's at minus 70 millivolts. Membrane potential just means like what is our electrical charge buildup on the inside of a cell compared to the outside. And the difference is minus 70 millivolts. So we say that our membrane's potential is minus 70 millivolts. So when we receive certain signals, our charge starts to go up. And if it reaches this critical threshold, right here at minus 55 millivolts, it will shoot up. This shooting up is called hypopolarization. It just means that we go all the way up. After zero, we start to get even positive charge. We peak at about plus 40 millivolts. And then we get into repolarization, which is that we have to lose this charge, this potential again, and we come back down. We go all the way even more down than this. So our resting is at minus 70, but we go all the way to minus 85 roughly. And then over time, we go back to minus 70. So this whole thing might take 1 millisecond or 3 milliseconds, like roughly that amount of time. And so that's about as fast as our neurons can communicate. So we can't send signals at more than 1 millisecond delays, which means that we can send at most 1,000 different signals from one neuron per second, usually even less. That's actually where brainwaves come in. And we'll discuss brainwaves a bit more in our final lecture, but basically it means how often our neurons communicate. Like typically we have brainwaves of like 15 hertz, which means that we communicate 15 times per second, which would be one communication every like 60 milliseconds or something like that. So neurons are mostly not active. They're only usually active a few times per second. These are just different values that we'll go back to. Don't worry too much about memorizing this, but the main idea is that the way neurons communicate, let me just bring up the chat right here. Yeah. Yeah. Never mind. Okay, so the way neurons communicate generally is that they build up this action potential, and when they reach up to this point, they shoot out a signal through their axon. Now and then they go back to relax again so that they are again able to get excited and shoot out. Now let's discuss what exactly shooting a signal means. All right, let's briefly discuss this. So neuron starts at minus 70 millivolts. It reaches this point of minus 55 millivolts, at which point it shoots up. And then after it has shot up, it needs to come back down and relax before it's able to send another signal. And this period is called a refractory period. And this is a duration during which a neuron is not able to send any new signals. This is usually 1 or 2 milliseconds, not much, but just it exists. Okay. Now let's talk about what exactly it means for a neuron to send a signal to another neuron. This is called a synapse. And let's have a look at what's going on here. So imagine this is the axon terminal of one neuron. So let's just briefly go back here. You can see that this is an axon. At the ends, we have axon terminal, and this will connect to another neuron's dendrite. So right here, this pink one is an axon terminal of one neuron, and this blue one is a dendrite of another neuron. And basically what happened is that in this pink one, an electric charge arrived all the way to the end. Like, let's see this restarting. Like right here, you can see this is an electric charge that arrives. And it causes some operations at the end of the axon, like opens a gate, some particles flow in, and neurotransmitters are released. And these neurotransmitters connect to the other neurons' ion channels, and they open up the ion channels that causes electrical charge to build up in this other neuron. Now, don't worry about not understanding all of this right away. I want to just show you visual intuition right now, and now we'll discuss it more slowly. Now, this is just the same process, but as an image so that we have more time to comprehend what's going on. So let's see. Right here or here we see a neuron, and this is the axon of this neuron, and this is what it looks like. Over here all the way, this is connecting to a dendrite of another neuron. This whole place right here where they connect is called the synapse. This thing right here is the axon terminal, like this round thing. So this is the end of the axon. And anytime the charge builds up inside the neuron, this charge then travels all the way down the spine and arrives here. The charge, when it arrives here, sends a signal that it's time to release neurotransmitters here. Now, what is here? Here is some space, and this space is called the synaptic cleft. This is where neurotransmitters are released, and neurotransmitters are just chemicals that will connect to ion channels on this receiving dendrite, and they will change how quickly charge builds up in this receiving neuron. So these places are called receptors. They're basically ion channels, and this is the synapse. So some terms to throw out now. These are just mitochondria, which is energy creating part of the cell. They're here because they need to drive some energetically expensive things. They don't worry about them right now. But this is called, so synapse has a presynaptic side and a postsynaptic side. Presynaptic side is the axon that sends the signal, and the postsynaptic side is the dendrite that actually receives this signal. So together they're called the presynaptic axon terminal and the postsynaptic dendrite. And together they make the synapse. So the axon terminals for synapses with dendrites or sometimes somata of other neurons. Somata is just plural for soma, which stands for the cell body, like what we see here. So an axon usually connects to a dendrite, but it might also connect to the soma itself. So let's just briefly go back to this. This axon might connect to another neuron, either a dendrite or directly to the cell body. Because regardless, it's going to just connect to ion channels, which exist both on the dendrite and on the cell body. It's just that they are usually on the dendrite much more. So what's going on here is that we send an electrical charge all the way through this. And when it arrives here, it gives the order to release neurotransmitters that are in these bubbles. And these neurotransmitters arrive on the dendrite, and they cause the buildup of charge inside the dendrite. So basically, what we see here, these increases are because some neurotransmitters arrived and they connected to our ion channels and temporarily opened them, which causes the charge to start to go up. So this is the main idea with synapses. And our video is ending. We will rejoin in a moment, and then we'll see a visual explanation of this from 2 Minute Neuroscience, which is a very good YouTube channel for short, brief introductions into all the neuroscience concepts. I highly recommend it. I will link a few of them in our Discord as well, which I think are relevant. But we'll just see their synapse explanation because I think it's very good. And this is the main idea, basically, that we have one neuron's axon sending a signal to another neuron's dendrite to cause it to spike either faster or slower. Okay, before our video ends, do you have any questions?
No.
Anna, and after all neurotransmitters restore, then we can start again. So basically, neurotransmitters connect.
Okay. Afterwards, in the video, there was a process where neurotransmitters, like, they transfer some particles, and after that, they disappear.
Oh, yeah. Okay, yeah, I get your question. We'll get to that in our lecture, but basic idea is some of those neurotransmitters are cleaned up, destroyed, and new ones are made simply in the axon. Some of them are reuptaken, which means that they return inside the axon, and some of them can even build up there. And yeah, basically, some of them are destroyed, some of them are taken to other places, and some of them return to the axon. But basic idea is that axon and just neuron keeps producing more of the neurotransmitters. And spiking is about release of the already produced neurotransmitters. And each neuron has different kinds of neurotransmitters. Like some of them you probably heard about, like dopamine, serotonin, oxytocin, and so on. They generally, they do different job depending on which brain area they are in. But like pleasure is mostly about dopamine, mm, serotonin is mostly happiness, and so on. And there's also adrenaline, which is for fight or flight kind of situations. But the idea is these are different kinds of chemicals which are released by the axon terminal inside this synaptic cleft space. And the dendrites that receive them will hold them for a while, and then eventually they will let them go. And once they are let go, they keep floating around, and some of them are cleaned up by the glia and other particles in our body, and some of them return inside the axon to be sent out again the next time a signal arrives. Uh, we'll see it visually as well in a moment, but yeah, that's the rough idea of it. Uh, does that answer your question? Okay. All right, let's continue our lecture. And yeah, if you get any questions, again, interrupt me. So, roughly, this is what a synapse looks like. We send out an electrical signal from the axon. This signal tells them it's time to release the neurotransmitters outside. And basically, we have all these bubbles inside. These bubbles are called synaptic vesicles. And they pretty much just hold a bunch of neurotransmitters inside them. And so when they receive the signal to go ahead and release it, these bubbles are released outside and they dissolve and all the raw neurotransmitters get exposed in the outside. And some of them bind, which means connect to the dendrites here. I mean, the ion channels on dendrites, and they cause electrical buildup. So generally, there are different kinds of neurotransmitters. Some are excitatory and some are inhibitory. Excitatory basically means that when they bind to ion channels, they increase the electrical voltage inside the cell. Inhibitory just means the other way around. So they either decrease the charge or they slow down the rate at which charge can be increased. Mostly they just, yeah, so either increase the charge or decrease the charge. Which is also why our brains are not constantly active. Because, like, if you think about it, if we only had the excitatory kind of connections, then neurons would never go out. Like this one, say, excited this other one, and this one excited this other one, and now they would remain excited forever. And our brains would be basically fried. That's sort of what happens in epilepsy. When there is too much excitation, every neuron activates at the same time, and there's not enough inhibition, which means that they are not told to stop activating. That's how people get seizures in epilepsy. And yeah, that's what would happen if we didn't also have inhibition, which means that sometimes some neurotransmitters that are released actually decrease the activity of the target neuron instead of increasing it. We'll get to that in a moment, but for now, let's see 2 minute neuroscience is a very good visual explanation of how this works, the synapses.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
So this is a really good channel that I very highly recommend. It's called Neuroscientifically Challenged, and they have this 2-minute neuroscience playlist. I actually recommend going through the entire playlist just slowly, but I'll tell you some of my top picks and send them in Discord because I really think that they are worth going through because they're very short and they give a pretty good visual intuition. Like they alone are not enough to understand neuroscience, but for specific topics, they're really good. Yeah, I'll send a link of this. Okay, now let's continue our lecture. And if you have any questions, just feel free to, I'm just going to stop because feel free to interrupt. Now, yeah, this one more thing that I want to cover. So the kind of synapse we were looking at here is called a chemical synapse. So neurons communicate to each other with synapses, but there are two kinds of synapses. There's a chemical synapse, which is what we see here, that at the end of an axon, the signal is received and it sends out the neurotransmitters, and these neurotransmitters then either increase or decrease the action potential of the other neuron. And there is another kind of synapse, which is called electrical synapse. An electrical synapse is a lot more simple kind, but it's not the typical synapse, so it's not covered as much. Typically, if you hear synapse, it pretty much always means chemical synapse, like the kind that we're seeing here, with neurotransmitters, axons, dendrites, and so on. Now, what is electrical synapse? Electrical synapse is when two neurons have cell bodies that are directly attached to each other. They're attached by these things called gap junctions. And these gap junctions can be closed or open. They're made of these 6 proteins that together connect neurons. Like right here, you can see, like for example, these are 3 neurons, and these 2 are connected to each other, these 2 are connected to each other, and these 2 are connected to each other. And these are the electrical synapses. And what this means is that, so as we discussed previously, neurons spike, so they send out a signal because electrically charged particles build up inside them. So when they share physical space like that, with openings like this, they pretty much build up these electrical particles at the same time. Because for instance, if there's 10 ions here and 3 ions here and 4 ions here, it will stabilize such that each of them will get equal amount of ions. Because they will flow between them like with diffusion. And because these doors are open, they will basically build up to the same amount of charge, which means that they will spike at the same time. So electrical synapses are used when a group of neurons has to fire at the same time. It's not as common as chemical synapses and not as widely useful, but they still exist, and so we need to know about them. So basically the difference is here, one neuron talks to the other by sending out neurotransmitters from the ends of his axon, and the other receives it at the dendrite. Like here you can see neurotransmitters are released. They bind to the ion channels. These ion channels now open up, and so charge builds up inside the neuron, and then the one might spike. But for electrical synapses, basically the cell bodies themselves are linked. And so the charged particles, like there is no neurotransmitter here, it's just the charged particles directly. They are shared, and so spike will happen at the same time in all three of these cells. Like, let's just go back briefly. This is the cell body, and imagine cell bodies of two neurons might be connected directly to each other. And in that case, they will build up charge at the same time. Is this idea understandable? Are there questions?
No.
Okay. If you get any specific questions, feel free to interrupt me. I will also link some of the 2-minute neurosciences other videos as well, because I think they're very useful. But I guess it's time to continue this conversation. So, how do we move? How do we speak? How do we do anything? All of those are done by this thing called neuromuscular junction. And this is a place that connects our neurons with our muscles. So what we're seeing here is compare just to this image. In a typical chemical synapse, we have an axon in which when a spike is sent, meaning when electrical activity is sent.
Sorry, I can't hear you.
Oh, sorry. Can you hear me now?
Yeah.
Right. So in a typical chemical synapse, we have one neuron which sends out an electrical spike through its axon. And when it arrives here, it gives the order to release these synaptic vesicles, which have neurotransmitters. And then the other neuron's dendrite receives these neurotransmitters, and because of it, increases or decreases its potential, which will eventually lead to it also firing and so on. This is a basic idea when one neuron talks to another neuron. And this is the case for interneurons. But when we have motor neurons, one neuron's axon is talking not to a neuron, but to a muscle. And that's what we see here. So this is called a neuromuscular junction because it's a neuron and a muscle. Yeah. So this is our axon, just as before. But this time, our synaptic vesicles again release a neurotransmitter. It's a specific kind of neurotransmitter called acetylcholine. And at the other end, we don't have ion channels and a neuron dendrite anymore. Instead, we have a muscle. A part of a muscle that's called motor end plate. And this motor end plate receives this acetylcholine neurotransmitter that we released. And basically, it causes the muscle to contract. Contract means, basically, contraction is the only thing that muscles can do. And that's how we can move. That's how we can speak, by contracting our throat and tongue and so on in this correct form. And that's how we communicate, engage with the world, and do everything pretty much. So briefly, every muscle, in this case, I mean skeletal muscles. Now let's just back up quickly. We have three kinds of muscles in our body. We have skeletal muscles that we use to move around. For instance, our hands, our legs, and so on. They're called skeletal because they connect to bones and they move the bones. We also have some cardiac muscle, which basically means our heart. And we also have smooth muscle, which is just muscles inside our organs. So for now, let's just discuss the skeletal muscles. And the way skeletal muscles work is that they are made of muscle fibers, these small parts. And muscle fibers receive their signals from neurons. Like right here, you can see these green ones are axon terminals. And they connect to some muscle fibers. And they tell them it's time to contract. And that's how our body knows to move. We'll cover this in more detail in our 5th week, where we have senses and movements that Nina will explain. But for now, this is the main idea. And that's how these neurons enter, that they send a neurotransmitter called acetylcholine, which gives us order to muscles to contract. And contraction in a correct order makes our hand move this way or this way or this way or this way or whatever we are capable of doing. So any kind of movement or any kind of speech that we can do is by a specific contraction of muscle groups. We have dedicated brain areas to figuring out which muscles you have to contract to move correctly and so on. Like if you think about it, it might seem obvious how we should use our legs to get to, let's say, a door. But if you think about it, there's actually hundreds of different muscles in your leg, muscle groups, I mean, like just muscle fibers, which have to contract in exactly the correct way and exactly correct timing so that you bend one leg and then move it forward, then bend the other leg. But obviously, you don't have to consciously think about this because mostly because of evolution, but also some adaptation during our life. Like kids are born not able to walk, but then they learn it quickly. Essentially, we figure out how to walk, how to move, and everything because we have a brain area here called the cerebellum, which translates our high-level command of, let's say, our forebrain here, the frontal lobe will say, I want to go to the door. The cerebellum will figure out the calculations of, okay, for that, I need to move my leg 10 degrees up, then I need to move my toe a little, and so on. And then our motor neurons execute these commands. We'll cover this in more detail in fifth lecture, but this is the main intuition for now. All right, let's briefly cover what we said about spikes one more time, and then we'll go into neurotransmitters. And the time that we'll have left at the end, we'll use for Q&A and to just go back through these topics. By the way, if I'm speaking too fast, tell me and I'll slow down or go through some topics again. Let's just take a moment. As of now, are there any specific questions? Let me see if there's some.
No.
Sorry?
No.
Do you have a question?
No.
Okay. All right. So again, one more time, what we covered. At rest, every neuron has inside the potential of minus 70 millivolts. And when it receives signals because of neurotransmitters or because of whatever that give it more charged particles, the charge inside builds up. And if it reaches a threshold, which is usually considered minus 55, but sometimes could be minus 45 or whatever, but it's generally considered minus 55 millivolts. If it reaches here, a lot of the ion channels will open up and will quickly depolarize. Go all the way up here to plus 40 or plus 50 or somewhere around there. Because of going up to here, it will also send out a signal through its axon, and so give neurotransmitters to whatever neurons it connects to. And then it calms back down, loses the charge, loses it too much, goes all the way to minus 85. In some neurons 75, but generally 85. Then goes all the way back to rest again. This is the main idea of a spike, and is again main unit of communication between neurons. All right, with that, let's start discussing some specific neurotransmitters that come to mind. So you probably have heard of the happy hormones, which are dopamine, oxytocin, serotonin, and endorphins. Endorphins are also known as adrenaline, by the way. Here. Let me just discuss it here. Basically, each kind of neurotransmitter that we saw before, like here, some neurotransmitter is released. Depending on the neurotransmitter and on the brain area, they do a different job. Like obviously, technically, they all do the same job of making a neuron fire more often or less often. Like it's exactly the same job. But because of their different chemical configuration and because of their different place where they're used in the brain, they affect different purposes. For instance, dopamine affects pleasure, motivation, and movement. Let's just briefly cover all the main ones. So we have adrenaline, which is for fight or flight situations. So when we are in stress or when we are very excited, they cause an increase in heart rate and blood flow, which makes us ready to run away or ready to fight or ready to at least be more aggressive or more alert. We also have noradrenaline, which is more for attention and for responding to things, and also similarly to stressful situations. Then you have dopamine, which is for pleasure, addiction as well, like any kind of addiction you can think of, gambling addiction or drug addiction or even scrolling addiction or whatever. is caused by basically hijacking the dopamine centers. So dopamine makes us want to do something, crave something, and addictions basically hijack it so much that our craving is beyond our control, and then we have to keep doing it just to calm down our cravings. There are ways to train out of these addictions, pretty much by strengthening your conscious control. And your conscious control is roughly your frontal lobe's activity. And so, yeah, there are things you can do about it. Serotonin is known for happiness, and also it's needed for sleep, and yeah, just a bunch of related things to that. There's also GABA, which is a main inhibitory neurotransmitter. So everything else we're looking at here is actually excitatory, which means that they cause neurons to fire more. But GABA is inhibitory. That means that when a neuron receives GABA neurotransmitters, its action potential decreases, so it's less likely to fire. Then there's acetylcholine, which is a neurotransmitter used by muscles, but also in thinking, learning, and memory, so depending on where in the brain it's used. There's glutamate, which is the most common type of neurotransmitter everywhere, but like for learning, memory, development, and whatever. And we have endorphins, which are for exercise, excitement, sex, well-being, euphoria, and for reducing the feeling of pain temporarily, which is what some drugs try to do, like with morphines and such. So these are the different kinds of neurotransmitters. And yeah, again, the same neurotransmitter in different parts of the brain does different jobs. But yeah, these are the main things that we need to understand about them. Let's briefly, I want to mention one thing as well, which is that I try to keep notes on neuroscience related topics on my website. You might find them useful. Like about neurotransmitters, I have a listing of how they are created inside the cell body, how they are released. It's too detailed for this lecture, so I didn't go through them too much, but if you get curious, you can read through this. I'll also link some 2-minute neuroscience videos about this. Yeah, for like specific neurotransmitters, we can find out some more details and such. And also just generally, if you want to know more about neuroscience on your own readings as well, I keep a knowledge graph of it here. You might find it useful, basically. I have some neuroscience notes about, for instance, neurotransmitters, but also like maybe spikes, also about synapses and such, and you could find this helpful for studying. We'll have some homeworks about this, but general idea is that these are spikes, synapses, and neurotransmitters together. They make up the structure with which we communicate in our brain. All right, that's pretty much it for today's lecture. Now, if there's any questions, I'll hear them, and if not, we can end the lecture and I'll send all the relevant links in our Discord. Okay, thanks, Anna. Anyone else, if you have any questions, feel free to ask them now, or if you get them, you can ask them later in Discord. But yeah, that's it. All right, then. Thanks. Okay, see you next time.
Goodbye. Thank you for this lesson. See you next time.
Bye.
Bye.